Swayam Prabha. Digital India. Educated India.
Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. Let's continue our discussion about the different hosts and what we have, but before we discuss further, let's recap what we have discussed so far. So, what we have discussed so far? So, in the previous module, we have discussed the introduction of, we have discussed about the structure of prokaryotic cell, we have discussed about the structure of eukaryotic cell. Within this, we have also discussed about the different organelles which are present in the eukaryotic cell, their structure, their function, as well as we have also discussed what is their relevance in contributing into the different activities which are important for the eukaryotic cell. Then we have also discussed about the crucial and significant differences between the prokaryotic as well as the eukaryotic cell, and these differences are important because once you know these differences, you could be able to exploit or you could be able to use the prokaryotic cell as well as the eukaryotic cell for downstream applications in the biotechnology, because depending on the applications you can choose the prokaryotic cell or the eukaryotic cell. Then we have also discussed about the difference between the plant cell as well as the animal cell, and as we discussed before, these differences are also important to know what are the limitations or what are the technological limitations you have, or what are the technical challenges you are going to face when you use the plant cell or the animal cell as a source of host for any type of biotechnology applications. And at the end, we have also discussed how you can isolate the different organelles in a eukaryotic cell. We have also discussed in the same thing, we have also discussed the isolation of the periplasmic fraction as well as the cytosol from the prokaryotic cell as well. And in this section, in this lecture, we have also discussed about the different techniques which you can use to isolate the different organelles present in a eukaryotic cell. These approaches or the techniques can be utilized even for the different not only for isolating the organelles, but also for isolating the different types of biomolecules which are varying in their sedimentation rate or which are varying in terms of their different densities. So, these are the different things what we have discussed. So, what we have discussed so far is actually enable you to understand the information or the requirement of the host which we are going to use in the biotechnology applications. Apart from these resources, the host cell is also depends extensively on the metabolic reactions which they are going to catalyze, and these metabolic reactions are being used for many purposes. So, let us see what are the different metabolic reactions are occurring in the host cells. So, what we have discussed about the prokaryotic cell, we have discussed about the animal cell, and then we have also discussed about the plant cells, and all these cells, all these host specific cells are requires the energy, and this energy they require to run the various metabolic reactions to drive its life cycle or lifespans, and to divide and grow from one cell to many more cells. And there are there are two different types of metabolic reactions which are happening in a host cells. These are called anabolic reactions or the catabolic reactions. These anabolic reaction, as the name suggests, these are called as the biosynthetic pathways. So, anabolic reactions are the reactions in which the new biomolecules are being synthesized, whereas the catabolic reactions are the energy producing reactions, which means in the catabolic reactions you are actually generating the ATP by the breakdowns of the biomolecule, and then this ATP you are using for running the life cycle or running the different activities within the cell. So, there are so these are the two majorly processes which are happening in any of these prokaryotic cells, any of these host cells, whether it is a prokaryotic cell, animal cell or the plant cell. So, let us discuss about these reactions. So, we will start with the glucose metabolism. So, in the glucose metabolism, as soon as the glucose molecules enter into a cell, it is being channelized into in a series of metabolic reactions, so that you can produce the ATP as well as the reducing equivalent, and all these reducing equivalent as well as the ATP is being used to produce the energy. So, the first metabolic pathway what we are going to discuss is called as the glycolysis. Glycolysis is operating in a in cytosol of the cell, whether it is the bacterial cell or the eukaryotic cell. So, the first reaction which is required to channelize the glucose molecules into the glycolysis is which is catalyzed by an enzyme known as the hexokinase. So, the hexokinase utilizes the glucose. It can utilize any 6 carbon sugar, and with the help of ATP and magnesium as a cofactor, hexokinase generates the glucose 6-phosphate, and then glucose 6-phosphate is being isomerized in the fructose 6-phosphate. So, irrespective of whether you have a glucose, fructose, mannose or maltose, all these sugar molecules are being accepted by the hexokinase to generate their corresponding the 6-phosphate. For example, in the case of fructose, it will generate the fructose 6-phosphate. In the case of glucose, it will generate the glucose 6-phosphate. So, if and after that there is a isomerase reactions, this isomerase will convert all these forms to the fructose 6-phosphate. So, if you remember, in this place you are actually spending 1 ATP to commit the glucose molecule into the glycolysis. Once the fructose 6-phosphate is generated, it is again getting phosphorylated on the other carbon, and then you are generating the fructose 1,6-bisphosphate. The enzyme is phosphofructokinase, and one other molecule of ATP is being utilized in this process. Once the fructose 1,6-bisphosphate is being generated, then the fructose 1,6-phosphate is being split by the aldolase in the two molecules. One is called glyceraldehyde 3-phosphate, or the other molecule is called as dihydroxyacetane phosphate. These two molecules, glyceraldehyde 3-phosphate or the dihydroxyacetane phosphate, are the isomers. So, these molecules can convert to each other by with the help of an enzyme known as phosphotriose isomerase. Irrespective of these the formation of any of these molecules, the glyceraldehyde 3-phosphate is being further processed to form the 1,3-bisphosphoglycerate, and the enzyme is known as the glyceraldehyde 3-phosphate dehydrogenase. And in this process you are generating, you are using the NAD plus NPI, and you are generating the first molecule of NADH. These NADH molecules are called as the reducing equivalent. They will be further used in the downstream reactions to generate the ATP. Then 1,3-bisphosphate glycerate is being converted into the 3-phosphoglycerate, and the enzyme name is the phosphoglycerate kinase. This means you are removing the one phosphate molecule from this compound, and this phosphate molecule is being received by the ATP to generate the ATP. So, since you are generating the 2 molecule of glyceraldehyde 3-phosphate, you are going to generate the 2 molecules of ATP. Then the 3-phosphoglycerate is being converted into 2-phosphoglycerate, which is actually just the change in position of the phosphate within the molecule, and then from this molecule you are generating the phosphoenol pyruvate. This is a dehydration reaction where you are going to have a loss of water, and the enzyme name is the enolase. And then from phosphoenol pyruvate it will be get converted into the pyruvate, which means again you are going to lose another phosphate molecule, and this phosphate molecule is going to be received by the ADP to generate the ATP. So, now if you see very carefully, you are having the multiple places. So, initially when you started with the glucose, you are spending 2 ATPs, one at this place, the other one is at this place. So, you are spending 2 ATP molecule to channelize the glucose into the glycolysis, but you are generating 2 molecules of glyceraldehyde 3-phosphate, and at the end the glyceraldehyde 3-phosphate is getting converted into the NADH, and this NADH is going to be into the electron transport chain, and within the electron transport chain it will going to generate the ATP, which means you have spent 2 ATPs in the initial 2 reactions, and with the generation of NADH you have produced the ATP, and in the second reaction you with the extraction of phosphate molecule from the 1,3-bisphosphoglycerate you have generated 2 molecules of ATP, and in the last step when the phosphoenol pyruvate is also losing another phosphate molecule, you are generating another 2 molecules of ATP, which means if you see the balance sheet of ATP production from a glycolysis, you will see that with the glycolysis you are gaining many ATP by spending 2 ATP in the first reaction. So, let us see the glycolysis, the balance sheet. The balance sheet is like this, as we discussed. In step 1 to 4, you are actually spending 2 ATPs, and then in the subsequent step when you are generating 2 molecule of glyceraldehyde 3-phosphate, and in the step 6 you are generating the NADH, and each NADH molecule when it goes for the electron transport chain it generates 3 ATP, which means from the 2 NADH molecule you are going to get the 6 ATPs. Then in the step 7 when there was a loss of phosphate you are going to generate 2 ATP, and in the last step when the pyruvate is being generated you are going to generate another 2 ATP. So, what is the net balance? If you take the single molecule of glucose, what you have generated is the 6 plus 2 plus 2, which is 10, and the 2 ATP molecule you have generated in the initial 4 steps. So, that you are activating the glucose, and that is being the consumption. So, at the end of the one glucose molecule when it is getting oxidized in the glycolysis you are generating the 8 molecules of ATP. So, when the glucose molecules enter into the glycolysis you can see that you are actually spending 2 ATP molecules, but in return you are getting the 8 molecules back. But as the glucose enters into the cell and the glycolysis operates, all these events are under the tight control. So, let us see what are the factors are controlling the glycolysis reactions within the cell. So, within the cell you have the transporters which are present on the cell surface, and one of the hormone which is called as the insulin, and the insulin hormone, when it binds to the insulin receptor, it drives the downstream event or downstream signaling, and because of that it actually it regulates the glucose metabolism within the cell. And how it happened is that in the first event the insulin binds to its receptor in the cell membrane, and that actually activates the insulin on the insulin mediated downstream signaling. Once the insulin mediated downstream signaling is activated, it actually upregulates the recruitment of glucose transporter, which is GLUT3 and GLUT4, and these transporters then goes on to the cell membrane, and they are available for the glucose transport. So, as soon as they are available on the plasma membrane, they allow the glucose molecule to be coming inside the cell, and as soon as these glucose molecules are available, they are being used, or they will be available for the glycolysis. But you can imagine that if the glucose level is going is very high and the insulin level is going down, then in that case all these events are going to be reversed. The glucose transporter which are present on the cell surface are being internalized into the vesicles, and then ultimately they are being going to be degraded to form into the endosomal system, and that is how the level of insulin glucose receptors on the cell surface is going to be reduced. Once the glucose receptors on the cell surface is going to be reduced, the entry of the glucose is going to be less, and as a result the downstream glycolysis is going to be less. Apart from this glucose mediated control, you also have the control at the within the insulin within the glycolysis reaction which are operating in the cell, and these regulations are mostly either by the feedback mechanism or by covalent modifications. So, in the at the enzymatic level you have two different types of regulations. One is called the covalent modifications and the other one is called as the allosteric regulations. So, let us discuss about the covalent modifications. So, in the covalent modification, the example is the pyruvate kinase. The pyruvate kinase which is actually taking up the phosphate from the phosphoenol pyruvate, and it is generating the pyruvate. And you can see that as the enzyme is phosphoenzyme or this pyruvate kinase could be present in two form. One is phosphorylated form, the other one is the unphosphorylated or the native form. So, the dephosphorylated form is more active, and whereas the phosphorylated form is less active. So, you can imagine when there is a low blood glucose, which means that it is going to reduce the activity of glycolysis. So, in this low blood glucose event, what happens is that the dephosphorylated pyruvate kinase is going to be phosphorylated, and it will generate the phosphorylated pyruvate kinase, which is going to be the less active. But when the blood glucose will go up, and in that process what will happen is the phosphorylated pyruvate kinase is getting converted back to the dephosphorylated pyruvate kinase, and that actually is going to be more active, and it is going to catalyze more enhanced action or enhanced level of the conversion of phosphoenol pyruvate to pyruvate. Apart from this regulation, which is actually a covalent modification, the fructose 1,6 bisphosphate, if you remember in the initial 1 to 4 reactions, fructose is being phosphorylated by the phosphorylated by another round, and that has generated the fructose 1,6 bisphosphate that actually is positively regulating the conversion of phosphoenol pyruvate to pyruvate, whereas if there is a sufficient quantity of ATP or the alanine is present inside the cell, it is negatively regulating, which means if you have enough amount of ATP in the cell, it actually inhibits these reactions, or it will block the conversion of phosphoenol pyruvate to pyruvate, which means if you have sufficient energy, there is no reason that you actually utilize the glucose which is present inside the cell through glycolysis. The other kind of modification which is called allosteric regulations. So, in the allosteric regulation is where the molecule is regulated in a different mechanism. So, every enzyme has the allosteric site, and in the allosteric site the substrate or the another molecule actually goes and bind, and that is how it actually modulates their activity. So, you can imagine this is that the fructose 6 phosphate is getting converted into fructose 1,6 bisphosphate, and this reaction is catalyzed by the phosphofructokinase. So, phosphofructokinase has the allosteric binding site for fructose 2,6 bisphosphate, which is neither a substrate or nor the product of this reaction, and because of that it actually modulates its activity. Similarly, the fructose 1,6 bisphosphatase, which actually going to drive the reverse reaction, is also been having the allosteric binding site for fructose 1,6 bisphosphate, and fructose 2,6 bisphosphate is positively regulating the activity of fructose phosphofructokinase 1, and that means if the fructose 2,6 bisphosphate is present, it will activate the reaction, or it will enhance the catalytic conversion rate of phosphofructokinase, and as a result you are going to have more level of the fructose 1,6 bisphosphate, which will eventually get converted into the pyruvate. Whereas, if there is a sufficient quantity, then the fructose 2,6 bisphosphate is going to be negatively regulated. So, at the same time fructose 2,6 bisphosphate negatively regulate or will reduce the activity of fructose 1,6 bisphosphatase, which means it on one hand it increases the activity of phosphofructokinase. So, that will increase the phosphorylation reaction. On the other hand, it will reduce the activity of phosphatase, and by doing so fructose 2,6 bisphosphate allosterically favors the formation of fructose 1,6 phosphate. Once the fructose 2,6 bisphosphate function is over, the fructose 2,6 bisphosphate get converted into fructose 6 phosphate, and that is how it actually its effects are going to be controlled. Now, let us move on to the next reaction, and next reaction, once you generated the pyruvate, that pyruvate will enter into the next catalytic reaction or next metabolic reactions. So, pyruvate, once it is generated in the cytosol, it will transported to the mitochondria. This transport is never been in the pyruvate form. The pyruvate get converted into the acetyl CoA, and then the acetyl CoA moved in to the mitochondria and catalyze the downstream reactions, and these reactions which are occurring inside the mitochondria are called as the Krebs cycle. So, let us see what are the reactions are happening inside the Krebs cycle. So, as we discussed, the pyruvate which is actually being generated from the glycolysis is getting converted into the acetyl CoA. The enzyme name is pyruvate dehydrogenase complex, and once this acetyl CoA is being generated, it is being used to catalyze the first reaction of Krebs cycle. As the name suggests, the Krebs cycle is being discovered by a scientist named as the Hans Krebs, and it is actually the multiple reactions which are being used to catalyze the different steps to generate the ATP as well as the reducing equivalent. So, the oxaloacetate which is being present combined with the acetyl-CoA in the first reaction. The enzyme name is citrate synthase to generate the citrate. The citrate is being utilized by the aconitase. It is a dehydration reaction. So, one water molecule is being lost, and that is how it generates and produces the cis aconitase. Cis aconitase generate the isocitrate. Now, this isocitrate is being used by the isocitrate dehydrogenase, and in this reaction the first time the isocitrate is converted into the oxalosuccinate and a one molecule of NADH is being produced. So, this is the first NADH which is being produced in the Krebs cycle. After the oxalosuccinate, there is a decarboxylation reactions and it loses its one molecule of carbon and it get converted into the alpha ketoglutarate. Alpha ketoglutarate is being converted into the succinyl-CoA and the one molecule of NADH is being produced. This is the second NADH molecule, and the succinyl and the enzyme name is alpha ketoglutarate dehydrogenase, and the another second molecule of carbon dioxide is being released. So, this is second molecule of carbon dioxide, this is the first molecule of carbon dioxide. Now, the succinyl-CoA is being converted into the succinate, and if you see very carefully in this step the GTP is being produced instead of ATP. So, the first molecule of GTP is being produced. Now, the succinate is being converted into the fumarate with the help of enzyme known as succinate dehydrogenase, and the another molecule of FADH is being produced, which is actually the first molecule of the FADH2, which is a reducing equivalent. So far we have generated in the Krebs cycle they has generated two molecules of NADH. Now, the one molecule of FADH2 and the one molecule of GTP. Now, the fumarate is getting converted into the malate with the help of an enzyme known as fumarase, and the malate is getting converted into the oxaloacetate, and the third molecule of NADH is being produced. So, this is the third molecule of NADH, which means you are generating the three molecules of NADH, one molecule of FADH2 and one molecule of GTP. So, at the end the oxaloacetate which is being generated again goes through this cycle and combined with the acetyl-CoA to generate the citrate, and that is how this is called as the Krebs cycle, because this is a cyclic reactions occurring one after other. How people know that this is a cyclic reaction? So, the Hans Krebs has done many experiments and ultimately he has figured out that this is a cyclic events occurring, because if you block any of these substrates and you does not allow, or suppose you inhibit the fumarase, you could be able to block the Krebs cycle or you disrupt the convergence of one product into another product. So, at the end what you are getting out of the Krebs cycle, what you are getting is three molecules of NADH, one molecule of FADH, one molecule of the GTP and the two molecules of carbon dioxide. Let us see what is the balance sheet of the Krebs cycle. So, in a balance sheet of one carbon Krebs cycle, you always remember that you have generated the two molecule of pyruvate from the one molecule of glucose. So, the initial reaction is that in the first acetyl-CoA is generated. So, you have seen that when the pyruvate got converted into the acetyl-CoA, it has generated the one molecule of NADH. Then in the step 3, when there was a generation of alpha ketoglutarate, another NADH is being generated. So, this is the first NADH, this is the second NADH. Then in the step 4, when there was a succinyl-CoA generated, Then also there was a generation of the reducing equivalents, and then in the step 5 there was a generation of GTP. 1 GTP is equivalent to ATP. So, that is how you can have the 1 ATP or GTP. Then when there was a generation of fumarate, you have generated the FADH. So, if one molecule of FADH, when it goes through the ATS oxidation, or when it goes for oxidation into the electron transport chain, it generates the 2 molecule of ATP. And then the last step, you generated the oxaloacetate, and that actually also given the 1 NADH. So, that is how you have generated the 3 ATP. So, let us see what is the net balance. So, net balance of one molecule of pyruvate molecule, when it got into the oxidation, you have generated the 15 ATP molecule, and in the glycolysis you have generated 2 molecules of pyruvate. That means, from 1 molecule of glucose you have generated the 30 ATP molecule, which means you have generated 30 ATP from the Krebs cycle, and you have generated the 8 ATP from the glycolysis, which means if you oxidize 1 molecule of glucose, you will get the total 38 ATP if all these reducing equivalent will go for the complete oxidation. Similar to glycolysis, the Krebs cycle is also under the tight control. It is being regulated by at many steps, by many enzymes and by different types of mechanisms. So, let us see what are these mechanisms to control the Krebs cycle. So, the Krebs cycle is always or mostly being regulated by a feedback mechanisms, where the substrates, such as for example, for the first reaction you are converting the pyruvate to the acetyl CoA. In these cases, the pyruvate, the ADP, NAD plus, coenzyme A, fatty acid, calcium, these are going to be increase these activities. So, if there is an increase in the level of NAD plus, or if there is an increase in the level of ADP, what this mean is that the cell is having the less energy, which means you need to run the Krebs cycle to produce the energy. So, that is why these factors are going to enhance the activity of, or enhance the activity of the enzyme, which will catalyze the conversion of pyruvate to acetyl CoA. Once the acetyl CoA will get will combine with the oxaloacetate to form the citrate. At this step also, the ADP, which is actually the significance of the loss of ATP or low level of ATP in the cell, is going to enhance or will activate these reactions. Whereas, if you have the large quantity of ATP, NADH, acetyl CoA, or the fatty acid, that is going to inhibit these reactions. Same is true for when you do a conversion of isocitrate to alpha ketoglutarate. That time also ADP and calcium are going to be the positive modulator, they will enhance the activity, whereas the ATP and NADH is going to inhibit or will going to do a inhibition of these activities. Same is true for the other steps as well. So, the Krebs cycle is mostly being regulated by the feedback mechanism, which if the ATP is being generated, you have large quantity of ATP, then the ATP will go and inhibit the reactions. But if there is a large quantity of NAD, or there is a large quantity of ADP in the cytosol, which will signify that the cell is undergoing the low energy state, then that actually is going to upregulate the activities of different enzymes, and that is how it is going to enhance the different reactions, and that is how you are going to see the more activity or more Krebs cycle running. So, let us continue our discussion about the Krebs cycle, and what we were discussing, that the Krebs cycle is under the tight control, and where the various substrate as well as the products are causing the the feedback inhibitions. And the question comes, why the Krebs cycle is so much tightly controlled. The reason behind the tight control of Krebs cycle is that the Krebs cycle is the central metabolic pathway. So, just after the glycolysis, which actually where the glucose is being break down and get converted into the pyruvic acid, the pyruvic acid get enters into the mitochondria, and then it actually enters into the central metabolic pathway, which is actually called as the Krebs cycle. So, in the Krebs cycle, the pyruvate which is being generated from the glycolysis is being processed further, and then the several molecules of NADH, FADH2 as well as the molecule of GTP is being generated. So, apart from the purpose of Krebs cycle to metabolize the carbohydrate molecules to provide the energy in the form of the reducing equivalent, whether it is the NADH or the FADH2, the purpose of Krebs cycle is also to coordinate with many other metabolic pathways. So, let us see how the Krebs cycle plays central role within the metabolism of an organism. So, as you can see, the Krebs cycle, it is receiving the pyruvate from the glycolysis, and then the pyruvate is getting converted into acetyl CoA, and this acetyl CoA is actually entering into the Krebs cycle, and then it is getting processed with the help of several intermediates, and with these intermediates it is producing the reducing equivalent as well as the free energy, which is in the form of GTP. And what you can see now is that the oxaloacetate. So, oxaloacetate is also been channelized with the phosphoenol pyruvate. The phosphoenol pyruvate can be converted back to the glucose, and it also can be used to produce the different types of amino acids. And so, as you can see, the Krebs cycle is indirectly connected to the phosphoenol pyruvate or the carbohydrate metabolism, as well as the amino acid biosynthesis pathway with the help of oxaloacetate. In an alternate pathway, the oxaloacetate is also being used to synthesize the aspartate and other amino acid, and these amino acids, the carbohydrate which is present in these amino acids can be used even to synthesize the purine as well as the pyrimidine, which means the oxaloacetate is, which is actually the part of the Krebs cycle, is playing central role in connecting the Krebs cycle to the amino acid pathway, the nucleotide pathway, and as well as the resynthesis of glucose molecules. Similarly, what you can see is the citrate which is being synthesized in the Krebs cycle is utilized for the biosynthesis of fatty acid as well as the sterol. Similarly, the alpha ketoglutarate is used to synthesize the glutamate, and the glutamate is ultimately producing the different types of amino acids which are glutamine, proline and arginine. And what you can see also is the succinyl CoA. So, succinyl CoA is another intermediate which is present in the Krebs cycle, and that actually can be a precursor for synthesis of chlorophyll, heme as well as the porphyrins. So, different types of iron containing proteins could be synthesized using the succinyl CoA, which is actually the heme bound proteins. So, as you can see, the TCA cycle is a central pathway. It is on one side it is converted to the amino acid pathway, on other side it is converted, it is linked to the nucleotide biosynthesis pathway, and on the other hand it is linked to the fatty acid synthesis. Alpha ketoglutarate is linking the TCA cycle to the another type of amino acid synthesis, and as well as the succinyl CoA is connecting the TCA cycle to the heme biosynthesis of heme or the porphyrins, which means that if there is a requirement of these intermediates, like for example, if there is a requirement of glycine or serine, the Krebs cycle can donate or can utilize or can channelize some of these oxoacetate molecule for the synthesis of glycine or the serine molecules. Similarly, the other intermediate also can be used to channelize the carbohydrate molecule which are present in the TCA cycle to some of these derivatives, which means the Krebs cycle can actually regulate the different metabolic intermediates which are being generated in the metabolic which are being generated within the cell, and it actually makes the tight control between these intermediates. And because of this important role which Krebs cycle play within the metabolism, the Krebs cycle is under tight control by all these intermediates, and also as well as the intermediate which are being present in the Krebs cycle itself. But these are the situation happens, and these are the things which Krebs cycle is doing when the oxygen is present, because Krebs cycle is producing the large quantity of NADH, 1 molecules of FADH and the GTP. These NADH and FADH which are being generated in the Krebs cycle are need to be oxidized in the electron transport chain. So, all these NADH and FADH are going to go to the electron transport chain, where the different types of electron receptors are going to receive these molecules, and then they are going to process the electron which is being they are going to process the NADH or FADH to generate the ATP, which means, and in this electron transport chain the oxygen is required. So, as a result, if there is a oxygen is present, the Krebs cycle will run these intermediates, and as well as there will be energy generation. But what will happen, there will be no oxygen in the environment, or there is a low oxygen in the environment. So, as we discussed, if there is a oxygen, the TCA cycle will run, and it actually will produce the carbon dioxide and the energy which is being in the form of ATP. So, if oxygen is present, the TCA cycle will run, it will generate the 3 molecules of carbon dioxide, and it will going to process the reducing equivalents to generate the ATP. But once the oxygen is not present, the pyruvate which is being generated from the glycolysis is being channelized into 2 different pathways. In one pathway, it is going to be converted into the lactate, and in this process the NADH which is being generated by the cell is going to be utilized to generate the NAD plus. In the other pathway, where it utilizes the 2 enzyme, one is called pyruvate decarboxylase, the other one is called as the alcohol dehydrogenase. It actually do the decarboxylation reaction first to generate the acetaldehyde from the pyruvate, and then from the acetaldehyde it generates the ethanol, where a 1 molecule of NADH is being used, and the NADH is getting converted into NAD plus. So, let us see, and these pathway which are operating in the absence of oxygen are called as the anaerobic oxidation. Let us discuss these 2 reactions separately and understand what is the significance of anaerobic oxidation in the metabolic pathway. So, let us start first the first reaction, which is the conversion of pyruvate to lactate. So, as we said, this pyruvate is being generated from the glycolysis. So, from the glycolysis you have utilized the glucose molecule, and then you have produced 1 molecule of pyruvate. In the absence of oxygen, the pyruvate will not enter into the Krebs cycle. Instead, the pyruvate will get converted into the lactate or the lactic acid. This path, and in this process the 1 molecule of NADH is being converted into the NAD plus, and the enzyme which catalyzes this reaction is called as the lactate dehydrogenase. These kind of reactions are occurring in the mammalian cells, as well as these reactions are occurring in the bacterial system or the prokaryotic system. So, in the bacterial system, what happen is that when the bacteria is growing under the anaerobic conditions, it cannot utilize the the pyruvate into the downstream biometabolic pathways, and once the pyruvate accumulates into the cell, that actually drives these reactions, and the pyruvate get converted into the lactic acid in the presence of the lactate dehydrogenase. One of the classical example of the bacterial species which is utilizing this reaction very optimally and and producing the lactate or the lactic acid is called lactobacilli. These lactobacilli are the bacteria which are responsible for generation of curd. So, these are the things which anyway we are going to discuss in a subsequent slide. Now, let us go to the second reactions. In the second reaction, the pyruvate is getting converted into the alcohol or the ethanol. In this you have the two different reactions which are coupled, and the first reaction is being catalyzed by the pyruvate decarboxylase. As the name suggests, it is actually going to remove one molecule of carbon dioxide from the pyruvate molecules, and that actually will generate the one molecule of acetaldehyde, and once the acetaldehyde is being generated, then the acetaldehyde is being processed by the alcohol dehydrogenase, and the one molecule of NADH is being used to produce the NAD plus, and ultimately the acetaldehyde is getting converted into the ethanol. So, in this process the pyruvate is getting converted into the ethanol, and these are the reactions which are very oftenly and occurring in the yeast, and that is how the people are using the yeast to produce the alcohol. And this so let us see what are the different intermediates, or what are the different what is the mechanism of the pyruvate to alcohol conversion. So, as I said, this is the two step reaction. In the first step, the pyruvate is getting converted into the acetaldehyde. In this, what happen is the pyruvate decarboxylase has the TPP or the TPP as a cofactor. So, TPP is present in the form of a carboanion, and that actually binds the first molecule of the pyruvate. So, the first reaction is the deprotonation of TPP to form the TPP carboanion, and that actually binds the pyruvate. So, it attacks on the pyruvate molecule, and that is how you are going to have the complex. Once this complex is formed, the one molecule of carbon dioxide is being removed, and that is how you are going to have the intermediate. This intermediate is then being stabilized by a resonance to generate this intermediate, and ultimately in the step 4 from this intermediate you are going to have the protonation to generate the methyl derivative of the TPP, and at the end the acetaldehyde is being generated. So, this acetaldehyde is being generated from these reactions, and the TPP is being regenerated on the enzyme which actually catalyzes the same reaction again and again. So, that is how the pyruvate is getting converted into the acetaldehyde following this kind of complicated reaction mechanism, which is the reaction which the enzyme which catalyzes this reaction is called as the pyruvate decarboxylase. Now, let us go to the next reaction. So, once the acetaldehyde is being generated, it is getting converted into the ethanol or the alcohol. So, and the enzyme which catalyzes this reaction is called as the alcohol dehydrogenase. So, alcohol dehydrogenase is a zinc bound enzyme. So, in the alcohol dehydrogenase, the active site zinc is actually binding the your acetaldehyde, and the it is taking up the so in the first step what happen is the substrate acetaldehyde is binding to the enzyme bound zinc with a coordinate bond, and then the NADH is also binding to the enzyme in the second step, and then in the third step there will be a transfer of hydride ion from the NADH to reduce the acetaldehyde, and once the acetaldehyde is reduced, it actually generates intermediate, and that intermediate is getting the proton from the water molecules, and the ultimately the NADH is getting converted into NAD plus, and the acetaldehyde is getting converted into the ethanol. So, let us recap again. So, then the reaction of acetaldehyde to alcohol is generated is catalyzed by the enzyme called alcohol dehydrogenase, which is a zinc bound enzyme. So, the active site zinc is in the first step, the active site zinc is binding the acetaldehyde and is also binding the NADH, and then there is a transfer of hydride ion. This transfer of hydride ion from the NADH to the acetaldehyde, and that actually generates a intermediate, and then this intermediate is getting the proton, or it is actually taking up the proton from the water, and with the transfer of proton it actually generates the alcohol as well as the NAD plus as well as the NAD plus. So, by doing this, pyruvate is getting converted into the alcohol. So, what is the significance of anaerobic oxidation? What you can see is that in the anaerobic oxidation, so anaerobic oxidation has the significance both for driving the cellular physiology as well as it has the industrial relevance. So, let us discuss first the cellular role in the cellular physiology. So, under the normal conditions or the under the oxygen in the presence of oxygen, the glucose is getting converted into the pyruvate, and the pyruvate is getting converted into the energy with the help of TCA cycle, which means when it is getting converted into energy, the NAD plus is getting converted into the NADH, where and in the presence of, whereas in the glycolysis also NADH is getting converted into NADH. But when there is no oxygen, this NADH cannot go for the oxidation, which means there will be a shortage of the NAD plus, and if there is a shortage of NAD plus, the ultimately there will be an accumulation of NADH in the cell, and if that happens, the NADH the cell cannot produce the energy, as well as cell cannot actually run many of the metabolic reactions where the NADH is required. So, to avoid these circumstances, because you definitely need a pool of NAD plus, so that you can actually run the metabolic reactions which are important for the cell apart from producing the energy. So, because of that, what happen is the cell is going with a loss making policies, where the pyruvate is getting converted into the lactate, and the pyruvate is getting converted into alcohol. And in these two processes, what is happening? You are actually generating the NAD plus, because you are utilizing the NADH which is already been preformed in the cell, and because of that you are going to generate a pool of NAD plus, and these pool of NAD plus can be used for the metabolic reactions or alternate metabolic reactions, where to the some extent until the oxygen is not available for the cell, and that actually helps to the cell to survive for a longer period of time, and also it actually allows the cell to the to withstand the stress which are being generated in the absence of oxygen. Now, let us discuss about the industrial relevance or the industrial importance of this anaerobic oxidation. In the first step, the pyruvate is getting converted into ethanol with the help of the carbon dioxide. So, there will be a removal of carbon dioxide. So, the classical example in this case is the yeast. We have already discussed that this reaction is very upregulated in the case of yeast. So, that can be used for two purposes. One, which we have already discussed, that if you trying to make the bread, what you have to do is you take the small amount of dough and then mix it along with the yeast molecules or the yeast organisms. So, what will happen is, because it is a dough and there is a anaerobic condition inside the dough, the yeast will run its glycolysis to produce the pyruvate, but it cannot mobilize that pyruvate to generate the energy in the Krebs cycle. Instead, it will run this reaction, which means it will run the pyruvate to alcohol conversion, and in this process it is going to produce the carbon dioxide, and once the carbon dioxide is trapped inside the dough, it will make the dough very fluffy, and that is how you are going to have the bread which is having the pores. In the alternate pathway, you can also convert the pyruvate into the alcohol, and that alcohol can be used for producing the different types of beverages. Similarly, in a other pathway, when the pyruvate is getting converted into the lactic acid, this is very often found in the case of another bacteria which is called as the lactobacilli. So, in the case of lactobacilli, you are actually converting the pyruvate to the lactic acid, and lactic acid is actually reducing the pH of media, and because of that it actually converts the milk to the curd, and that is how the lactic acid bacteria or lactobacilli has a relevance in converting the milk to curd and production of the curd. So, the anaerobic oxidation is having a very huge industrial relevance. We have only discussed the two examples. One is the production of ethanol or the production of the bread, the other one is the production of curd. But apart from these two pathways, there are many pathways which are actually being found in many other different organisms to generate the different types of metabolites, and these metabolites have a very high economical values in the industrial context. So, what we have discussed so far, we have discussed about the cellular metabolism in the mammalian cells, and what we have also discussed the control as well as the relevance of these metabolic pathways in terms of driving the different types of reactions, as well as in terms of running the life cycle of the particular organism. We have also discussed about the anaerobic oxidations and its relevance in sustaining the life of the organism, and at the end we have also discussed about the industrial relevance or the physiological relevance of these anaerobic pathways. So, with this we would like to conclude the lecture here, and we will continue our discussion about the growth media as well as the different types of nutrient media which people used to grow the prokaryotic cells as well as the eukaryotic cell in the subsequent lecture. Thank you.